Alright folks, so this is a video where we're going to be comparing some cells. You're going to see a bunch of different versions at different zooms, and so we're going to start off with some bacteria cells called Heliobactone pyloni. We can see zoomed out what that looks like and a little bit closer in. And we're going to zoom all the way in and see what that looks like all the way in. Now take a look at some animal cells, some salamander liver cells. And so again, zoomed out, medium zoom, and all the way in. And we can start to really see some things when we're all the way zoomed in there. And another single-celled eukaryote, like Paramecium, this time this one's called amoeba. And we can see medium zoom here. It's a little hard to see anything going on there, but when we get a little zoomed more in, we can start to see some organelles. Those are the little slides that we just used there. We're going to take a look at some human cheek cells from my own mouth, and we're going to dye them blue so that we can see the organelles inside. And so you can see that slide I've prepared, and here you can see some cheek cells. At the medium zoom, you can start to see that there's more than one cell there, and all the way in you can actually see some organelles on the inside of those cells in that big clump. Take a look at some plant cells, some onion cells, nice and thin so we can take a look, and again we're going to dye it blue so we can maybe see some of those organelles showing up. If we don't dye it blue, it doesn't really show up as well, and you'll see that in the next example. And so all the way zoomed in, we can see that little blue mass. Wonder what that is. And take a look at some onion cells with no dye, and you'll see how you can't really see the onion cells quite as well, and you definitely can't see that mass on the inside, which is really important. That's why we always dye these cells so we can see their organelles on the inside. And I didn't dye the celery this time, but we'll take a close look at some celery as well. You can see how I peel it nice and thin, and you can see it under our microscope. Halfway zoomed in there, and we'll zoom all the way in here, and we can see some features of those cells, and we're going to take a closer look with the still images